Welcome everyone to the 17th seminar series of RadNet, City of London. I am a senior RadNet postdoc working with Professor Kerbenodi-Valadilk at the Barts Cancer Institute and Eric Sahai at the CRIC. And my project is around investigating radiotherapy-derived endocrine signals from tumor blood vessels and how these endocrine signals modulate the tumor microenvironment and thus responses to radiotherapy. So upon my studies, I came across Dr. Michael Tsabar's papers on p53 dynamics and the work of Professor Galit Lava on p53. And so today we welcome Dr. Michael to present his work. So just a few housekeeping notes. So please mute yourself and turn your cameras off during the presentation. And then at the end, just pop your questions on the chat or raise your hand and I'll give you the speaker opportunity. And this meeting will also be recorded and for people who aren't able to be here today to be able to watch it later on. So we welcome Michael and thank you again for agreeing to do this talk. Michael will be discussing his research. So he's done his PhD on molecular and cell biology at the Brandeis University. Sorry if I misspelled that. And then he moved on to Professor Galit Lava's lab for his postdoc. And the Lava's lab uses live cell imaging to study dynamics of signaling networks in response to various stimuli with the focus on the dynamics of the tumor suppressor p53. He's been studying heterogeneity in p53 dynamics in response to ionizing radiation. And he's also working on developing a new system for connecting p53 dynamics with single cell transcriptomics. And so he will present both these works today. And so Dr. Michael has been first author in a number of high impact factor publications around p53 dynamics. So we look forward to hearing his talk today. Thank you. And I'll stop sharing so you can start sharing your screen.
Thank you. Right. Is it okay? Everybody sees?
Yeah, looks fine.
Okay, great. So thank you, Rita, for the kind introduction and for giving me the opportunity to present my work here today. As Rita mentioned, I'm a postdoc at Galit Lava's lab, which studies the dynamics of signaling networks, how they're controlled and what outputs they give, with a particular focus on the transcription factor tumor suppressor p53. The work I'm going to share with you today shows how escape from ionizing irradiation, DNA damage induced arrest, results in a divergent of p53 dynamics that may contribute to non-genetic heterogeneities in cellular responses to irradiation, which is a particular interest of mine. Sorry. Yes. Okay, so I'm going to start with a relatively brief and rudimentary introduction that I think most of you will find pretty straightforward, just to help me coin some terms before I dive into the project, and hopefully get a chance to touch on some of the difficulties that we came across while trying to address different fates that are resulting from different dynamics and how we plan to address these challenges in the future. So as many of you may know, the DNA in our cells is exposed to many lesions on a daily basis that must be reconciled in order to maintain genomic integrity. In this very crude schematic and somewhat juvenile, if forgive me, this mother cell following mitosis gives rise to two identical daughter cells that will continue to proliferate normally and carry on cellular functions like the mother. However, if the DNA in the mother is damaged prior to replication, several outcomes can take place. In the first and probably most advantageous, the DNA is repaired prior to replication, and the cells proliferate normally, resulting in two identical daughters that would carry on cellular functions like the mother. However, if the DNA is not reconciled, the DNA damage is not reconciled prior to replication, the cells can either be eliminated from the cellular pool by terminal cell fates, such as senescence or apoptosis, or, and probably the least desired outcome, the cells can continue to proliferate with perturbed cellular functions, such as the controls on proliferation itself, which can lead to the onset of cancer. So to allow for sufficient time for repair to take place, the cells employ the highly conserved DNA damage checkpoint that is governed by the two PI3-like kinases ATM and ATR that phosphorylate a cascade of downstream effectors, the most important of which, from our perspective at least, is the tumor suppressor and transcription factor p53 that will regulate cell cycle arrest, also known as the guardian of the genome. So what is p53? p53 is a transcription factor that regulates the transcription of several hundred genes in response to various stresses. Interestingly, these genes can belong to somewhat contradictory transcriptional networks. For example, it's relatively difficult to reconcile why a cell that has activated apoptotic pathways would also try and repair the DNA concomitantly. So to understand or to reconcile these contradictory roles, we must understand how specificity is embedded within the p53 signaling network. There are many ways to confer specificity in signaling networks. As I mentioned before, the Lahav lab studies specifically protein dynamics as a layer of specificity in signaling networks and in the signaling network of p53. And when we say protein dynamics, we mean any temporal changes that occur on the protein following a stimulus. And this can be the localization of the protein. It could be post-translational modifications, changes on the proteins. And today we're going to talk about the temporal changes in protein levels themselves following ionizing radiation, which I'm going to term p53 dynamics from here on in, just for simplicity's sake. We can't discuss p53 dynamics without mentioning the negative feedback loop p53 forms with its own E3 ubiquitin ligase MDM2. So when a stimulus is sensed, in normal conditions, p53 levels are kept almost at zero just to prevent cells from responding. But when the stimulus is sensed, p53 levels go up, the p53 stabilizes, and that results in accumulation of MDM2, its own E3 ubiquitin ligase, which would degrade p53, resulting in loss of MDM2 because p53 levels are now low. And if the stimulus persists, such as the damage, p53 levels will go back up, resulting in accumulation, reaccumulation of MDM2, resulting in loss of p53, loss of MDM2, et cetera, et cetera. And you can kind of envision how this negative feedback loop forms this seesaw behavior that should result in a pulsatile behavior of p53 levels, protein levels following stimulus. And we can actually visualize this pulsatile behavior if we tag p53 with a fluorescence reporter. This is, in this case, it's a venous reporter, and treat the cells with ionizing radiation. You can see that p53 is mostly localized to the nuclei, as we expected. And after we irradiate the cells, the levels of p53 go up and down in the nuclei in a very striking way, at least to my eyes. Sorry. So if indeed the dynamics confer specificity, we should be able to show the different dynamics result in different cell fates. So that's exactly what Jeremy Purvis did in his seminal science paper from 2012, where he showed that damages that result in accumulation of double-stranded breaks, such as ionizing irradiation, are associated with these pulsatile dynamics that we've seen, these oscillations of p53 that we've seen. And we can actually see these also when we look at a Western blot. We can see the first pulse at around 2 hours and second at around 7 hours. And these are associated with cell cycle arrest and pro-survival outcomes of the cells. However, damages that result in accumulation of single-stranded DNA, such as exposure to UV, lead to a sustained p53 dynamics, as can be seen here in the Western blot on the right. And these are associated with terminal cell fates, such as apoptosis and senescence. Jeremy went one step further, and he wanted to show that it's not the type of damage that leads to the divergent cell fate, but it rather is the dynamics. So to do this, he used Nutlin 3A, which inhibits MDM2, thus allowing p53 levels to accumulate. And when he radiated the cells, we can see on the left, when cells are not treated with Nutlin, they again give these nice pulses. However, this very carefully curated regimen of Nutlin treatment resulted, even after ionizing radiation, with accumulation of p53, and that was associated with accumulation of senescent cells as in the sustained dynamic as compared to the pulsatile dynamic, showing really that it's the dynamics that are important and not the damage that the cells encountered. So we started this project that I'm going to share with you today with a relatively naive question, which is up until that point, most of the studies that were done in the lab really focused on the establishment of this feedback loop and looked very carefully at the first hours to a day, like 24 hours of response to ionizing radiation and to stresses. We just wanted to know how long would these pulses persist? Would they go on indefinitely, or would something else happen later on? So we again looked at p53 levels following ionizing radiation, as you can see here on the left, this Western blot that shows this first pulse at 2 hours and second pulse at 8 hours. That is a result of this negative feedback loop that we've discussed between p53 and MDM2. Now you can notice that the first pulse, the levels of p53 at the first pulse are much higher than the levels of p53 at the second pulse. And we hypothesize that this is a result of a gradual loss of synchrony between the cells. So the cells are exposed to the treatment all at the same time, and the first pulse is then therefore quite synchronized to when it occurs. However, slight differences between the cells should result in a gradual loss of synchrony, and what we hypothesized which should be a leveling off of p53 levels between somewhere between the trough that we see at 5 hours and the peak that we see in 2 hours. We were therefore quite surprised to see that when we plot for p53 levels at later time points, 48 and 72 hours, we see a reaccumulation of p53 levels to levels that actually exceed those seen at the first pulse. And this indicated that there is some reactivation of p53, and we were wondering if this is basically how penetrant this phenomena is. Is it something that occurs in all the cells and is relatively gradual, or is it something more dramatic that occurs in only a subset of the cells? So to address this question, we turned back to our basically bread and butter, which is live cell imaging of MCF7 cells, which are a breast cancer cell line, where p53 had been tagged with a Venus reporter and irradiated the cells with 10 gray ionizing irradiation or 10 gray X-ray. And you can see these pulses that we've seen before, and you can actually see this gradual loss of synchrony that I've discussed that kind of fit our hypothesis. But later, the later phase cells, actually a subset of the cells, seem to do something quite different and accumulate p53 levels to a much higher extent as opposed to their environment. And we can look at the data in a little more quantitative way using these heat maps in which every row represents one cell that has been tracked throughout these the experiment. So here we have about 460 cells, give or take. The x-axis represents the time that elapsed from irradiation, 10 gray in this case, and the p53 levels are shown in this color map where p53 with low p53 levels are in blue and high p53 levels are in yellow. And we sort the cells on mean p53 intensity, and you can notice just by looking at this heat map that a subset of the cells show a marked late phase increase of p53 levels as compared to the entire population. We call these cells switchers because unlike most of the cells that remain oscillatory throughout the experiment, an example of which can be seen here at the top, the switching cells start off by being indistinguishable from their oscillatory counterparts. However, later on, in this case around 36 hours, the cells switch to something that looks a little more like a sustained dynamic. Then I wanted to know if the fraction of cells that switch correlates to the amount of damage that the cells sense. Or in other words, can I cause more or less cells to switch by changing the dose of irradiation? So I repeated this experiment with an increasing dose of ionizing irradiation from 1 gray to 20 gray. And just by looking at these heat maps, I think you can appreciate that initially we do see this positive correlation between the dose and the level of switching up until 5 gray, where if we irradiate the cells further, we actually cause a decrease in the fraction of the cells that switch. We can separate the switchers from non-switchers in an unbiased way using k-means clustering. You can see that it does a pretty good job at separating these two populations. In the shaded area, you can see that 50% of the cells around the median. The median is shown here in this highlighted line, and you can see in the bar graph at the bottom the fraction of cells that switch at every dose, and you can see indeed the first increase up until 5 gray that we hypothesized is a result of increasing damage, but we were quite perplexed as to what prevents cells from continuing to have this correlation between dose and switching. Basically, what force acts counter to the accumulation of damage and prevent cells from accumulating more switching at higher doses. And thankfully, around the same time, Jose Reyes from Galit's lab published a paper that shows that fluctuations of p53 amplitude can lead to escape from ionizing induced arrest, and the fraction of this, the cells that escape is inversely correlated to the dose of damage or the dose of irradiation that the cells are exposed to, which sort of makes sense if you consider the fact that the higher the dose, the more stringent the DNA damage checkpoint is, preventing cells from escaping the arrest. So then I wanted to know if the cells that switch actually escape the arrest. Basically, are the switchers escapers? The first hint of this came from closely observing the movies. And those of you who paid close attention and are familiar with live cell imaging probably noticed that the switchers tend to have accumulation of micronuclei following the switch. So here you can see the mitotic event, and after the mitotic event, you can see that p53 levels go up, and we can notice these micronuclei. And as many of you may know, micronuclei are a hallmark of mitotic catastrophe. So that indicates that these switchers actually manage to progress through the cell cycle. So indeed, when we looked at how many cells divided in the switching versus non-switching population following irradiation, whereas most of the non-switching cells remained arrested throughout the experiment, as seen here in blue, most of the switching cells, about 80% of them, managed to escape from the cell cycle induced arrest and progress through the cell cycle at least once, or mostly once, following the irradiation. Moreover, the division time and the switching times are somewhat well correlated, with division time preceding switching time by a few hours. So this, I think, indicates that division and switching, or progression through the cell cycle and switching, are relatively well correlated, but I wanted to know if there is also a causal effect between progression through the cell cycle and switching, or in other words, does switching require the cells to progress through the cell cycle? To address this question, I used CDK4/6 inhibitor, ribociclib, which arrests the cell in G0 and G1. And if we treat the cells with ribociclib prior to irradiating them, we prevent the cells from escaping completely. As can be seen here in this bar graph, all cells remained arrested throughout the experiment. So if progression through the cell cycle is required for switching, these cells that have been treated with ribociclib should not be able to switch, which indeed they don't. So if you look here at the heat maps, we can see the fraction of switchers in the control in the 10 gray, and cells that have been treated with ribociclib don't switch at all. The converse experiment is to try and persuade cells, if you will, to escape more readily from the DNA damage induced arrest by knocking down the main effector of p53 that controls the G1S arrest, CDKN1A or P21. So I hypothesized that if we knock down P21, we should see more cells progress through the cell cycle, and which indeed is what we see. So if we knock down P21, we see about 70% of cells manage to escape from the DNA damage induced arrest. And similarly, these cells show a much higher fraction of cells that switch as compared to the control counterparts. So I hope I managed to convince you at this point that p53 dynamics can switch from oscillatory to sustained dynamics in a subset of cells following irradiation, and that this progression through the cell cycle is required for switching. Interestingly, also, one other thing that we notice is that the maximum switching occurs at mid-range of irradiation, which perhaps proposes an optimal damage to checkpoint intensity ratio. So if the DNA damage checkpoint is too strong, the cells will not switch. But if it's too weak, there will just be not enough damage to cause the cells to switch. Then we wanted to know what is the molecular mechanism that regulates this accumulation of p53 at the later phase, or the switching. And when we talk about the response of p53 to ionizing irradiation and double-stranded breaks, the usual suspect is ATM, which controls p53 activity or response to double-stranded breaks. And if we inhibit ATM 1 hour prior to ionizing irradiation, we lose p53 response completely. So because the switching is a late-term event, or late phase event, we wanted to see if inhibition of ATM 16 hours following ionizing radiation would prevent the cells from switching while allowing for the initial response of p53, the oscillatory response of p53 to take place. We can see here that this inhibition does a very good job at preventing CHEK2 phosphorylation, which acts as a proxy for ATM activity. So ATM is indeed inhibited by our treatment. However, the switching still took place, as can be seen here in the Western blot. Similarly, if we inhibit ATR 20 hours following irradiation, we can cause a loss of CHEK1 phosphorylation that is a proxy for ATR activity, but we can still see this switchers that we can see here in the ATM. So this caused us to conclude that the switching is independent of the canonical DNA damage checkpoint that is known and published to activate p53 in response to damage. And it kind of left us scratching our heads trying to find potential targets, because there aren't a lot of other acceptable activators of p53 in response to damage. Thankfully, at the time, Galit had an interaction with Professor Trudy Oliver from the University of Utah that pointed us at the direction of the caspase-2 pitisome as a possible regulator of p53 activity. The caspase-2 pitisome is a heterotrimeric complex comprised of PID1, or p53 induced with death domain 1, RIDD, which is a scaffold protein, and caspase-2, which interestingly is one of the most, if not the most, conserved caspases, but its roles are somewhat controversial. Thankfully, Trudy, when she was a postdoc at Tyler Jax's lab, showed that caspase-2 mediates the cleavage of MDM2. More recent work on the caspase-2 pitisome showed that it actually responds to a variety of mitotic abnormalities, such as failure of the spindle assembly checkpoint or accumulation of supernumerary centrosomes. And this positions the caspase-2 pitisome in a very comfortable place to actually respond to this escape from the DNA damage-induced arrest and degrade MDM2, thus resulting in accumulation of p53. So the first thing we wanted to know if MDM2 depletion is sufficient for switching. To show this, we turned to previously published computational model of p53 oscillations that takes into account various attributes of p53 rates and shows these oscillations. And we added a component of the pitisome that would degrade MDM2 in response to the mitotic event. And you can see that in our computational model, this degradation is sufficient to result in this switching, as can be seen here in this red graph. The next question is, okay, so our computational model is satisfied with the degradation of MDM2, but is it really the pitisome that degrades MDM2 leading to the switching? And if it is the pitisome that leads to switching, the perturbation of the pitisome should prevent cells from being able to degrade MDM2 and prevent, therefore, the cells from switching, keeping this negative feedback loop between p53 and MDM2 intact, and continuous oscillations should ensue. So we turned we basically perturbed the pitisome via two parallel channels. One of them is a CRISPR-Cas9 knockout of PID1, and the other is an inhibitor of caspase-2 that was recently published and showed to be very effective and potent. And in both these cases, the perturbation of the pitisome led to the complete loss of the switching fractions, as can be seen here in the heat map at the top and at the bottom. This is the PID1 knockout, and this is the caspase-2 inhibition, and the quantification of which can be seen on the right. So you can see that there is almost a complete loss of the switching fraction in both these treatments. Conversely, overexpression of PID1 was sufficient to increase the switching. So we overexpressed PID1 using a doxycycline inducible PID1 construct, added dox at 24 hours following irradiation, so to allow for the initial response to be normal. And we show that simply by overexpressing PID1, we led to a marked increase in the fraction of the cells that switch from about 15% to about 50%. So we conclude from this that the switch in p53 dynamics is independent of the canonical DNA damage checkpoint and is governed by the caspase-2 pitisome. So to sum this part up, we, I think, come to a realization that there are two phases of the response of the cells to ionizing radiation. In the early phase, the cells encounter the damage and establish the p53 control circuit between p53 and MDM2, leading to cell cycle arrest and resulting in these oscillatory dynamics that we see. Now, if the cells fail to repair the damage, then there are two choices that they face. The one choice occurs when the DNA damage checkpoint is continually satisfied and ATM and ATR continuously maintain the arrest. The DNA damage the cell cycle arrest will be maintained and this oscillatory dynamics will persist. However, if the DNA damage checkpoint fails to maintain the G1S arrest, the cells will progress through the cell cycle, and that would result in the basically activation of the caspase-2 pitisome, the degradation of MDM2, and this switching event that would result in a sustained p53 dynamics. So why do we think cells switch? And I'm going to start by saying a little letdown. Currently, we don't have any data to support our hypothesis as to why cells switch, and I'm going to share with you what challenges we have when we try to collect such data, and what are we doing to resolve these problems now. However, our hypothesis currently leans on several observations. In one, the first observation is that cells switch following failure of the canonical DNA damage checkpoint, which we have established, and that sustained p53 dynamics were previously shown to be associated with terminal cell fates. So we want to propose that switching could act as a failsafe for the canonical DNA damage checkpoint, or in other words, the caspase-2 pitisome may act as a failsafe for ATM and ATR for when they fail to maintain the continuous arrest. And this could be in a cell autonomous way by upregulating terminal cell fates, such as been seen before, but it can also act in a systemic way by inducing innate immune response programs and signaling to the innate immune response participants to clear these switcher cells out of the cellular pools. Interestingly, the switchers have an accumulation of micronuclei that have in recent years garnered a lot of attention in their ability to activate innate immune response via the SIGA-STING pathways. Another thing that could occur in these switching events, and that kind of maybe leans a little bit towards a possible therapeutic aspect of these switching events, is that micronuclei have been shown to induce an abscopal effect. And basically, the more micronuclei are accumulated, the more cells, the more tumors in the environment respond to treatment, which could also open up basically other therapeutic avenues and perhaps indicate where we need to be on our treatment regimen. Again, currently we don't know, and I'm very hesitant in making big claims around therapeutics using cell line studies, but it could be an interesting avenue to explore. The challenge that we have when we try to attribute cell fates to dynamics in a heterogeneous dynamic landscape is the way by which we try to establish fates. So we can take an imaging approach to establish fate, which of course allows us to track the dynamics of the cell, but is somewhat restricted in the time scale. We can only image the cells for so long before they start misbehaving. And the amount of imageable fates is relatively restrictive. So we can know, for example, if the cell died or not, but it's harder to know if a cell died because of apoptosis or necrosis or other pathways without developing specialized markers. We can know if a cell is arrested, but it's harder to know if the cell is senesced and what type of senescent that is without developing additional markers. The alternative is to use molecular approaches such as single-cell RNA sequencing, which is of course much more precise, gives us breadth of data, which allows for higher predictive value, but sadly mostly is incompatible with live cell imaging. So in order to know what transcriptomes occur in each cell, we need to forego the dynamic information, and of course, that would pose a problem when we try to associate dynamics to fate. So what we want to be able to do is to take a plate in which cells have been treated with a stimulus, in our case, ionizing radiation, and every cell has a heterogeneous dynamic, and be able to we believe that to be able to associate the different dynamics to the different fates, we must first understand the transcriptomics that are associated with every particular dynamics, especially considering that p53 is a transcription factor and its dynamics it brings about different cell fates, probably by regulating different transcriptomics. And this is what I'm going to discuss up till the end of the talk, basically how we're planning to address this challenge of bridging these two worlds apart. So in the last couple of decades, the combination of computational tools with live cell imaging allowed us to basically develop a new approach towards studying behavior of cells, which we term single cell live cell imaging, but really what it is live cell imaging with single cell computational analysis. And this allowed us to really appreciate the heterogeneity in signaling responses, such as you've seen today, but also this is from a study from Andrew Peck from Galit's lab that showed that cisplatin can lead to fractional killing, and this is associated with the rate of accumulation of p53. This is the dynamical landscape that I see of the cells in response to cisplatin, but this is not unique to p53. In the bottom here, you can see NF-kappa B's response to lipopolysaccharides from Marcus Govert's lab. You can see the heterogeneity there. Heterogeneity in responses has been seen in BHLHs, in ERK signaling, and basically almost every signaling network that has been studied with these live cell imaging and computational tools showed a relatively high degree of heterogeneity. In parallel to this, of course, the development of Single-cell RNA sequencing really exploded our understanding of the heterogeneous nature of the molecular states of cells in differentiation, in response to stimuli, and in almost every different attribute of the cell. And the evidence to this is the current human cell atlas that really shows heterogeneity beyond our imagination 20 years ago. Unfortunately, these two fields, as I mentioned, really developed in parallel. So sequencing and dynamics have really developed in parallel and with very few means by which to bridge the gap between them. And this is particularly unfortunate considering that most signaling networks bring about changes by regulating transcription. So in the center of most signaling networks lay transcription factors, and differences in dynamics should really account for differences in transcriptomics and subsequent different fates. So there are several ways by which various labs are trying to bridge this gap currently. I'm not going to go into any of them. Suffices to say that none of them were quite compatible with what we are trying to do. So we have collaborated with Andy Yoon's lab from MGH that developed these wavelength encoded laser particles, or basically tiny wafers seen here as black dots in these images, each of which is encoded with a particular wavelength between 1200 and 1600 nanometers. So each one is a unique snowflake, 1 out of 400, allowing us to separate 400 cells. So of course, if a cell uptakes more than one of these disks, that's 400 in the power of 2, 3, etc. So you can kind of see how this can be very highly multiplexed. These disks are readily engulfed by the cells for whatever reason and have very little effect on cellular behavior, basically no observable effect that we've noticed so far, and no changes to their transcriptional landscape. So the workflow that we're working on currently would allow us to first incubate the cells with these disks, then image the cells as we've imaged before, with the disks in them, basically. At the end of the imaging experiment, take them to a spectrophotometer that will allow us to associate a combination of disks that are present in each cell. So basically allowing us to tie the dynamics to a particular disk combination. Then we sort the cells into a 96-well plate, then scan the plate again with a spectrophotometer, and basically assign different optical barcode, different disk combination to each and every well. So that plate will then be analyzed with SmartSeq 2, allowing us to have the transcriptomes for every particular well, so allowing us to attach a transcriptome to an optical barcode, and we'll have the dynamic attached to the optical barcode, and this will allow us to orthogonally analyze both dynamics and transcriptomics and to see how they relate. We have basically solved the first half of this workflow currently. This is how the image looks after the spectrophotometer. Each of these color dots represents one disk. Upon closer or zoomed in image, you can see that some cells have 2, some cells have 5. So and some disks are free floating. And Nicola, the postdoc I'm collaborating with, is now able to send me these coordinates that are associated with different lasing frequencies. And then I can take these coordinates, find the cell in our images, and track it and analyze its dynamics. We are now currently working on solving the sorting aspect of it that proved to be a little more challenging than we first predicted. But this, we hope, will allow us to really intricately study the relationship between dynamics and transcriptomics, and not only allow us to further appreciate how different dynamics are associated with different cellular outcomes, but hopefully will also allow us to develop markers that will increase predictability of cellular behaviors in the long term from a very short-term observation such as imaging. So with this, I'm going to end, and I'm going to thank the Lab, in particular, Galit, for being a fantastic mentor scientifically and otherwise, which is not less important. Ashwini, our lab director, which has been instrumental to a lot of aspects of this and other studies. Caroline, Vina, Jose, Kyle, and Jacob all contributed to this study, and all members of the Lab. There are a wonderful group of people. I think I see Alina here. Really smart, hardworking, and fun environment to be around. Aviv Regev was my co-advisor before she left to bigger, better things in Genentech. Trudy Oliver is our wonderful collaborator on this. Peter Sorger and Laura Malesowski of the Lab of Systems Pharmacology, the American Cancer Society for my funding, and you guys for your attention, for inviting me, and for allowing me to share my work with you. And I'll be happy to take any questions.
Thank you, Michael. That was absolutely great to hear you. So maybe I can kick off with one question. So I was wondering, do you know what happens to those p53 persister cells? I know you said you don't have this completely clear, but are those cells, even though they are progressing through the cell cycle, are they the ones that are going to be senescent? Are they the ones that are going to be resistant to the therapy? So what's your idea?
It's a great question, and we've taken several approaches trying to address this, and none of them was very fruitful. But I will say that in our hands, MCF7s following 10-gray irradiation, most of them will be senescent if we just take into account beta-gal, which, you know, you can, and you take it with a grain of salt. Beta-gal is a very biased approach towards senescence. So most of the cells are senescent. Interestingly, and this is anecdotal, I've not done a lot of statistics on it, the cells that have managed to escape and switch are cells that tend to have a lower beta-gal accumulation, which sort of makes sense, because I think what happens is that these cells fail to senesce prior to basically a particular time frame that they have to senesce in order to escape from the cell cycle. So I think there is a time frame by which the cells decide what's going to happen. They try and repair the damage, they fail, and they try and senesce to maintain this arrest. If they fail to senesce, I think what happens is that they escape and they cause this accumulation of micronuclei. And really, I think, and this is an active avenue of research in the lab currently, that these cells are trying to signal to their environment to be cleared out. I think mostly they will stay put. They will not divide again, but they are trying to basically say, like, help me, take me out of here. I'm done. I'm deadly. Mm-hmm. When we look at lower doses, and again, this is something that we haven't really had a lot of emphasis on, but at lower doses, these cells tend to divide less. So they would, after they have this accumulation of micronuclei, they don't often divide anymore. And the reason we think is that their DNA is just scrambled beyond repair, and they just can't satisfy the spindle assembly checkpoint again. But this is a little marred by the fact that cells in lower doses do manage to repair. So the fraction that manages to repair really skews our study, our statistics, because you divide it 3 or 4 times.
Yeah, thank you. That's great. So we have a question from Lucy. Thanks for a lovely talk. The fluorescent disc technology is fascinating. I was wondering what the dynamics are of the uptake and retention of the discs.
Right. So it's a fantastic question, and I've one of my small contributions to the development of this technology was to really work on the uptake protocol. So the dynamics of uptakes are really quick in our cells, at least. And I've tried A549s, RPEs, and MCF7s. RPEs, I haven't tried a new protocol actually, but what happens is that you basically just need to incubate them for I'll be happy to share the protocol with you, but you need to incubate them for 15 minutes, 37 degrees on a rotisserie. You incubate 5 discs to 1 cell ratio, and then spin them down, resuspend, repeat the 15-minute incubation. By 30 minutes, most of the cells will have discs in them, and there will be very few free discs. The retention issue is a little more tricky. So they retain just fine. If the cells don't divide much, then they will stain the cells throughout the experiment for 72 hours and longer. The problem becomes if they do divide, then the division or the distribution of discs is stochastic. So some cells will have discs, some cells won't have discs, and if you had 5, they can go 3, 2, and there is really no way of really knowing where they're going, which is an issue if you want to track them for the experiment, but it's not that problematic if you just want an endpoint. You don't really care. So that's, I hope that kind of answers the question, but the cells are very happy to keep them inside. They really have no problem keeping them for the duration.
Great. So we have 2 more questions from Priyanka. So great talk. First, what is the difference in the oscillatory pattern p53 switching in relation to p53 mutations? This is because there is differential response to radiation based on the mutational status. And second, what is the relation to RB1 in the context of G1S phase?
These are both really good and fascinating questions. The dynamics, if I understand correctly, the dynamics of the mutation of p53 mutants is something that we have tried to study. Actually, Alina that was here can talk much more about it because she was the one trying to decipher it. Mostly what we see with mutants of p53 is that they have no oscillatory dynamic, and just an accumulation of p53. Actually, in many of the mutants, the amount of p53 is through the roof. And this is mostly because we think that they don't bind DNA as well. Most of the mutants of p53 is in the DNA binding domain, and therefore they don't activate MDM2 that much. And MDM2 will not degrade p53. So they just have a lot of p53 in the cells, which may or may not be inert. In some cases, we know it is not inert, and it has gain of function attributes, and in others, we really don't know. But we did try and study this a little more carefully, and I think this is sort of a back burner project at the moment. I don't have a good answer to question 2, and I think it's really fascinating. I don't know the status of RB1 in the context of the G1S arrest. I do know that, excuse me, that as I've shown, that when we knock down P21, we basically prevent the cells from arresting. So RB1 may play a role there in the fraction of cells that manage to remain arrested, like 30%, which is not insignificant. But I don't quite know what it does for the cells that do manage to escape when we knock down P21. And so it's a great thing to continue and study. I mean, I know it's not satisfactory, but that's what I have.
Okay, do we have any more questions? Okay, Jamie, let's see. Yes, go ahead.
Hi, Michael. Good to see you. Yes, I have a question relating to your like non-monotonic function of, or switches as a function of dose, where you get an increasing number of switches and then a decrease again as you go to higher doses. Have you been able to look at like longer movies? Because if it relates to like escape from cell cycle arrest, could it just be that the ones that you irradiate with 10 and 20 gray just haven't had long enough to be able to like exit arrest, and you'd find just as many switches or more in those as if you were able to image them for a bit longer?
Well, the short answer to this is no. It's something that was on my mind to do for a long period of time before the pandemic hit, and then we were scrambling to publish this and not do any more work. I think I actually think it is a fascinating question, because I do think, if I'm allowed to hypothesize. So as I said, the answer is no, but I do think, and this is stemming a little bit from studies that I've done in yeast with the DNA damage response, that there is a window by which the cells are allowed to have a mistake basically in the DNA damage checkpoint. So I think, or allowed to repair basically. The cell and the cells will have, I've tried to look at this statistically a bit, and I think there is a fixed error rate within the DNA damage checkpoint. And what this error rate is, I currently don't know. I try to put a value to it, so you can kind of envision, if you push the dose higher, if you see a constant fraction of cells that manage to escape from arrest, you can maybe make an argument as to, okay, to the error rate of the DNA damage checkpoint is about 5%. But to do this, as you mentioned, you really need to push the movies a lot further, because there is a relationship, as you so wisely hinted to, between when the cells escape and the dose that they've suffered. So cells that escape at G1, at 1 gray, usually escape within 18 hours. Cells that escape at 10 or 20 gray usually escape within 36 to longer. So it's not a linear relationship, but there is a relationship. And really, I mean, it's a great movie to do. The problem is a bit, and this is how we kind of, explain this to our reviewers, is that there is no real known endpoint. So in essence, you really want to visualize, to find the long tail, you need to visualize it indefinitely, which obviously technically you can't do.
Yeah.
But I do think it definitely merits another try, and looking at them for a week at least to see, because it's also possible that everyone switches, right? It's possible that the 10% of you switch at 10 gray, they're actually 100% after a week. I don't think that's, I will be surprised if that's the case, but I do think it's not a possibility that we can easily discount.
Okay, thanks. And do you mind if I ask one more slightly more tangential question?
It's up to Rita, if that's okay.
Yeah, go ahead.
Thanks. So you mentioned Jeremy's science paper where with the using Nuttallin to change the dynamics. So one thing I found interesting about that paper is that the UV treated cells, most of them die by apoptosis, right? But the ionizing radiation ones, once you change the dynamics, most of those become senescent. So what are your thoughts on why the cell fates are still different, even though the dynamics now look similar?
Right. I think it's a wonderful question, and we've been asked this a lot. And the short of it again is that we don't know. We do think that the type of damage still matters. So UV would cause accumulation of single-stranded DNA, which usually signals through ATR. And ATR is essential, unlike ATM, which is non-essential. And it's also, of the two, if you look back at simpler model organisms such as yeast, in yeast, the equivalent of ATR, MEC1, is again essential and also has very distinct roles that are independent of the checkpoint itself. So it has a lot to do with the DNTP rates in the cells and turnover, which maybe indicates that there is something in the background going on in the UV treated cells, which is actually why we had to develop the Nuttallin treatment. So we do think that something else is happening in these UV treated cells. We don't know if it's p53 dependent. We do know only that the UV treated cells, if you look at the paper, the UV treated cells have an accumulation of Bax and Puma, which are pro-apoptotic. The irradiated cells have accumulations of YPL and other pro-senescent programs. So there is a difference still in the sensing there, despite the fact that the dynamics are similar. And it could be that something else is happening. It could, the other possibility, less so going into the model that we've selected, is that MCF7 cells don't have caspase 3. So it could be that all of them are trying really hard to, activate the apoptosome, but just failing because of the absence of caspase 3. And it perhaps interesting to repeat this study with cells that have a more intact death pathway, such as A549s. In fact, we do know that in A549s, if you irradiate them too much, they start dying. Unlike MCF7 cells, which you can blast them with 80 gray, which I've done, and they'll be, fine mostly.
Okay, great. Thanks a lot, and great to see you.
Great to see you too.
Thank you, Michael. If I may sneak in another question, I know we're running out of time, but so I was wondering if you tested any kind of different confluences of the cells or even compression, or if you've explored like co-cultures with other cell types, and does that change p53 dynamics? I know it's a very big question.
No, you have just you've asked a fantastic question that actually has to do a lot with two separate projects that are going on in the lab currently and are really, really fascinating. I don't know if Alina is still here. Where can I see it? Actually, if I but
I'm here.
Hey, so do you want to take the confluency question?
Yeah, go ahead.
Okay, so Alina is actually studying the behavior of p53. She started by studying the behavior of p53 in spheroids because, just to be closer to actually what happens in the cells. And we noticed that the more confluent the cells are, the less they oscillate. And this has something to do apparently with the age of the media, but also with contacts. And Alina has some ideas and hypotheses that she's testing, and I think it's really fascinating. We do know from other work that we can see oscillations when we sort of oscillations when we take mouse slices, but it's unclear as to the density of the cells. And I think that this is something that Alina will probably publish soon, and you'll be able to read about. But there is a relationship, an inverse relationship between oscillations and confluency. The other question of incubation with other cell types is very interesting, and I've hinted towards this when I've discussed the innate immune response as an active field of research in the lab currently. And we are trying, another postdoc, Paula, which I don't know if she's here, is trying to look at the relationship between natural killer cells and p53 oscillations and p53 behavior, basically. So, for example, one can hypothesize that the switchers would be cleared out by NK cells more readily than the non-switchers, which we currently don't know, but again, this is something we are trying. I will say, though, also that this disk technology that I mentioned at the end can also work for co-culturing and incubation. So you can envision a situation where you want to co-culture several types of cells, and each one gets a particular disk. Instead of sacrificing a fluorescent marker for it, each one gets a disk frequency, and you can kind of multiplex the amount of cells that you can image. All right, I hope that addresses that question.
That's great. I look forward to reading Alina's paper. So I think we don't have any more questions that I can see. So yes, I think we'll finish for today. So thank you again, Michael, for your time and your great talk and for sharing your stories with us. And thank you everyone as well for participating. And we look forward to the next RadNet seminar. Thank you. Bye-bye.